The 2018 Commonwealth Innovation Forum, brought to you by Destination Gold Coast.
We're going to talk briefly about feeding the people of the Commonwealth. And ladies and gentlemen, to do this, it is my absolute pleasure to introduce you to Distinguished Professor James Dale AO from the Institute of Future Environments at QUT. Thank you.
Well, thank you, Madonna. Certainly a pleasure to be here. I'm going to talk a little bit differently from some of the other speakers previously. I'm certainly going to talk about feeding the Commonwealth, but I'm going to bring it down to a micro level. And I'm going to talk about micronutrient deficiencies and overcoming micronutrient deficiencies. It's an area that we've been working in for quite a significant period of time. So micronutrient deficiencies are rated one of the third most important public health problems worldwide after HIV, AIDS and malaria. They disproportionately affect developing countries, and I'll show you in a minute, they disproportionately affect Commonwealth countries as well. And the big ones are vitamin A, iron, zinc, iodine and folic acid. This is vitamin A deficiency, and this is what I'm going to concentrate on this morning. And as you will see, okay, in red are the countries where there is clinical vitamin A deficiency in at least 15% of the children's population under 5. And if you look at that, it's sub-Saharan Africa, it's South Asia, a little bit of places like Papua New Guinea. When you think about these countries, most of them, or the majority of them, are actually Commonwealth countries. So the majority in the region with South Asia, of course, with India and Pakistan and Sri Lanka, coming across here, Nigeria, down to East Africa, Uganda, Kenya, and then we go right down to South Africa, Malawi, Botswana, etc. Huge impact across the world. So particularly across the developing world. The outcomes for vitamin A deficiency, death is obviously the worst one, blindness, night blindness, but more importantly we now understand that there's impaired immune deficiency, impaired immune system development and impaired brain development. So massive impacts. On average, every year, around about 1 million kids under 5 either die or become permanently blind from vitamin A deficiency. This is a condition we know exactly what causes it and we know exactly how to fix it. And yet we have this incredibly bad outcome. So there are various ways of overcoming micronutrient deficiencies. The first one, food aid, it's very good in the short term, it's unsustainable. The two big ones that have had probably the most impact have been supplements and food fortification. There are two major problems with these two strategies. One is that for food fortification, you've got to buy food. And for supplements, you have to be close to a health clinic. In most of the countries that we're dealing with, they don't buy food. There's a population that don't buy food, they only produce their own food, and they're nowhere near a health clinic. So these things fail, and it's around about 15% of the population in developing countries fail to be impacted by supplements and food fortification. The other concern is what's known as donor fatigue. And donor fatigue is donors have been giving money for a long time to the same thing. They really want to spread their money. They like a good bang for their buck as well. And for these two strategies, it's every year, year in, year out. And so we now have a problem with donor fatigue funding these ongoing costs. Changing diets and crops, not particularly successful. There have been some examples where we've had minor changes by getting subsistence populations particularly changing their crops and diets. More recently, it's been this concept of biofortification of staple foods. It's a relatively simple concept, and that is to provide the micronutrients in the food that these people eat. And these are what we call the poorest of the poor. We can do it by two ways. One is by conventional breeding, and the other one is by genetic modification. Conventional breeding is great if the genes are available in the gene pool that's used by the conventional breeders. That's often not the case. And where it's not the case, then we can turn to genetic modification. And by far, the best example of biofortification, of genetic modification, is golden rice. Golden rice has been around for a long time. It's by far the most advanced of the genetically engineered crops and this is of course provitamin A. But it's had a whole lot of problems on the way through, particularly getting patent or freedom to operate has been one of the major ones. It's getting much closer to fruition, and hopefully in the next 12 to 18 months we'll actually see the release of golden rice to be grown commercially by farmers. I should mention that in Australia, golden rice has been approved as a food, not for growing, but as a food very recently by our Office of the Gene Technology Regulator. Okay, so what are these staple crops? Well, everybody, or virtually everybody in developing countries can pick the first four: corn and maize, wheat, rice, potatoes. Relatively straightforward. Many people, of course, think potatoes come from Ireland and get a real shock when they find out that actually from South America. Okay, if you then ask people, what about these other five? Forget about soybeans, it's primarily an industrial crop. What about the other five? Most people can't identify those because these aren't crops of the Western world. Cassava, primarily Africa, but some Asia as well. Bananas and plantains, people never get that as a staple crop. Maybe sweet potatoes, but certainly not sorghum and yams. So these are the crops that we need to target to be able to develop food biofortification. I'm going to talk about bananas, and often people confuse bananas and plantains. Plantains are just a subset of bananas, so we can lump those two together quite easily. When we look at who produces bananas around the world, it's also quite an interesting list. By far the biggest producer of bananas is India. China is now the second largest, but it's going down for particular disease. These two have around about 1.3 billion people. The country where we're working is Uganda. Okay, so it's number 3 on the list, 9.5 million tonnes of bananas every year for a population of 40 million. So you can understand that the consumption of bananas in Uganda is way above what you would expect in India and China, or elsewhere for that matter. It's very, very high. Okay, so I'm going to talk about Uganda, and I'll expand that a little bit later. But there are major micronutrient deficiencies in Uganda, and these are both iron deficiency anemia and vitamin A deficiency. These are the figures we got from the 2006 health survey in Uganda. There are more recent ones. They're not fully analysed yet, so I can't put those up. But what I can say is the situation is actually deteriorating rather than getting better. So when we look at vitamin A deficiency, between 15 and 30% of children under 5, and same for women of childbearing age, have clinical vitamin A deficiency. The low number is in the cities, particularly in Kampala. The high numbers are out in the rural areas. Iron deficiency anemia is even worse. So up to 80% of kids under 5 have clinical iron deficiency anemia. Just some photos here giving you an idea of bananas in Uganda, very different to what we see when we go down to Woolworths or Coles to buy some bananas by the hand maybe, or probably even singly. People in Uganda buy bananas by the bunch. And the consumption across the whole country is around about half a kilo per person per day. And that's everybody in the country, average. Okay, so in 2005, the Bill and Melinda Gates Foundation, under the Grand Challenges in Global Health, proposed Grand Challenge number 9, which was to create a full range of optimal bioavailable micronutrients in a single staple plant species. So that was 2005. They funded 4 projects in this Grand Challenge. One was ours. I should say everybody had to have a name because the Golden Rice Group had come up with golden rice, so everybody had to have a name. So we called ours Banana 21, which is actually just bananas for the 21st century. There's a cassava project and also a sorghum project. The sorghum project is no longer funded through the Gates Foundation. So that was a very important moment in time where a major foundation started to fund these projects across 4 of the major staples in the world. So we had bananas, and our target country was Uganda, and it said around about ½ a kilo per person per day. Bananas are their staple food. Bananas are their starch source. These are bananas that are harvested green and they're cooked for 3 or 4 hours, and then they're mashed, and it's sort of like a mashed potato. It's called matoke. Bananas are called matoke. Oops, I've gone a bit far. Bananas are called matoke, and if you go into somewhere to eat in Uganda and you say you want food, you'll get bananas, because food and matoke are the same word. They eat the bananas known as East African Highland bananas. They're consumed as a mash, and these bananas are low in both provitamin A and iron. This is what it looks like when this was staged for us, where women in beautiful local costume, but these are peeling bananas going into a pot, and then they're steamed for 3 to 4 hours. And they eat these bananas 3 times a day: breakfast, lunch and dinner. The project we had, and we decided right from day one, was a fully collaborative, integrated project where in Australia we did all of the technology development. And there were 2 reasons for that. One, we were set up for it, and the other one was that there is a real concern, particularly in developing countries by developing country governments, of being manipulated. Not just genetic manipulation, general manipulation. So the idea was everything we did in Australia first. We took it through Australian regulations, we took it through Australian field trials, we did the nutritional studies and stewardship outside Uganda. And then what we did was to transfer the technology, but not the plants, transfer the technology to Uganda, our collaborators, the National Agricultural Research Organisation of Uganda, and the transformations there, bananas and analysis field trials, ultimately are being done in Uganda. And the concept was, right from day one, these bananas were going to be made in Uganda, and that's been a very important part of our strategy. I should mention that what we put into bananas is not vitamin A or retinol. It's actually provitamin A, which is a combination of alpha and beta carotene. These are orange, okay, so it's very important. Provitamin A is converted in the human liver or animal liver into vitamin A or retinol. The most important thing is you can't overdose on provitamin A. You can't overdose on alpha and beta carotene. You might go slightly orange, but you won't get any toxicity. Whereas vitamin A, you can get toxicity in high levels. Because we will have no control over the level of consumption of these bananas, we had to go for something which was completely non-toxic. Okay, so these are the timelines. I'm going to go through these fairly quickly. So funding in 2005, and we had to determine our target level. What level did we want to reach in those bananas to know that we were being successful? Okay, we had to take into account a number of factors. Firstly, we had to know what the estimated average requirement for vitamin A was. We knew that was from other studies. Alpha and beta carotene are converted at different rates from different matrices, so we needed to understand how efficient alpha and beta carotene in bananas was to retinol, so vitamin A. So we could work out the conversion factor, and we actually took a very conservative conversion factor. Daily consumption sorts of numbers we knew. The interesting one was doing the processing and the processing losses. So at one stage our lab smelt more like a kitchen than it did a laboratory. But we worked out that around about 75% of the provitamin A was lost during steaming. So at the end we came up with a number, and this is 20 micrograms dry weight equivalent, or dry weight of beta-carotene equivalent. So just remember the 20. That's the important one. Okay, this is a metabolic pathway for going down to alpha and beta-carotene. And I mentioned earlier that golden rice, they started about 10 years before us. And the strategy they finally used was to target two enzymes or three enzymes in this particular system from geranyl diphosphate down to alpha and beta-carotene. These are the key enzymes. This one is a sort of a crossroads molecule. It goes off to all sorts of things. But going from there down to there was going from geranyl diphosphate down to lycopene was the absolute key. So what golden rice has done is has taken a phytoene synthase, this PSY gene from maize, but also a gene from Erwinia called CRIT-I, which actually substituted for these two enzymes here. So obviously when we started, we said, okay, well that worked for golden rice, we better add that into our list of things that we were going to try. However, rice, naturally occurring rice, doesn't contain, the seed doesn't contain any alpha or beta carotene. The whole pathway is shut down. Not so in bananas. So this is our Cavendish banana. Remember our target was 20 micrograms per gram. Cavendish contains around between 1 and 2, so it's very, very low. There are bananas with very, very high levels, and one called Asapoena comes from Papua New Guinea, contains somewhere between 100 and 150 micrograms per gram. Pretty exciting stuff. So we thought, yep, this is clearly one that we should be looking at as a potential source of genes to go into our bananas. East African highland bananas, they're around about half the level that we wanted. And this one is probably really important because this is a new banana coming through. It's an East African highland banana type, but it has disease resistance. It's even lower again. So we had to take both of those into account. So what we needed to do was to identify the genes and decide how we were going to find out which was the best of those. We also pulled out this banana phytoene synthase gene out of our Asapoena. We recognised right at the beginning that we didn't actually know what was going to work. But bananas are very big. They're much too big, and the numbers of light far too great to assess in a glass house. So we did something quite. It was a gamble. We went to our Office of the Gene Technology Regulator and said, what we want to do is we want to take these bananas, we want to genetically modify them. We've got a group of about 20 genes, we've got a group of about 20 promoters. We want to mix them all up. We want to try all the different permutations and combinations. We don't want to characterise them in the glass house. We want to go straight to the field. And then we held our breath. And believe it or not, they said bananas are about the safest crop you could do genetic modification with because they're sterile. So there's no transgene flow. And so they came back and said, these bananas are big and they're sterile. There's going to be absolutely no transgene flow. So away you go. They gave us a permit to do that. And so we went straight to the field, which was, I think, previously unknown. So in Australia, we went with Cavendish because it's genetically very similar to East African highland bananas, so we were able to use that as our model crop. Just to give you an idea, you want to make sure that you get things right when you work with genetic modification of bananas. If we do a transformation here on what we now call embryogenic cell suspensions, before we get any plants, it's about 12 months. We then need to multiply those and acclimatise them, so we're up to 18 months. Then they have to go into the field and we have to get fruit. That's another 12 months, so we're up to 2.5 years. And by the time we do the analysis, it's 3 years. You don't want to get things wrong here. You want to get as many things right at day one, because when you find out here you made some dreadful mistake, it's way too late. So what we did is we then went into our first field trial in Australia in 2009. It was in North Queensland. It's a place called South Johnson, just near Innisfail. One of the reasons we went up there is that it's in the tropics. We could plant bananas all year round. We had 2 field trials. We started with 14 different plantings and nearly 2,000 lines of bananas that we took into the field. This is what the field trial looked like. If you looked on Google Earth, this is the field trial here. This is the South Johnson River. This was probably flooded over the last couple of weeks with those dreadful floods up north. And we used Cavendish. So that was very important. Okay, so we went through a whole lot of iterations to identify what was going to be the best combination of promoters and genes to put into bananas. So this is from our final field trial. I won't go through all of the permutations and combinations that we went through. But this one, this line here, 324, which is, for those that are interested, a constitutive promoter, but this is the banana gene. And the banana gene turned out to be way, way better than anything else. The other important thing, if you go back to what I said about golden rice, it took 2 genes, one from maize and one from a bacteria. We only needed 1, and we only needed 1 gene from bananas. Our target, remember, was 20 micrograms per gram. This one was over 50 micrograms per gram. So we were way above target. There were many others that were also above target. So here's our wild type down here. It was about 5 in that. So plenty of others were way above target, but this was really incredible. So we knew at that stage that we were able to do it. And importantly, we were able to do it without what's known as yield drag. And that's what they look like. So that's a wild type Cavendish banana there. And this is our line 324 with a single banana gene. So really bright orange, these fruit. They look absolutely spectacular when you see them. So that was an important part. So we then had, we'd made that final transgene selection. We'd selected out which gene we were going to take, and we actually went with two different constructs. And then the next bit, so this is again going back to that biochemical pathway, metabolic pathway. So we just took a gene from this banana, this is Acerpina from Papua New Guinea. We took a single gene to add to that phytoin synthase that was already there in bananas. Okay, so then we started the whole program over in Uganda, and that was to transform East African highland banana. We went for one particular type called Nakatembe, and also the M9, this resistant hybrid that had been developed. So this was a very important part of the program, obviously. So as I mentioned, M9 is a new conventionally bred cooking banana with very significant disease resistance. And in Uganda, disease resistance is extremely important. Nakatembe, our East African highland banana clone, and we actually took two different constructs over, one with a banana promoter and one with a constitutive promoter, and then went and generated a whole range of events that we then took through to our elite line selection trial, holding our breath the whole time as to whether the technology was going to be able to transfer from Australia into East African highland bananas in Uganda. And lo and behold, yes, they did. So this is the field trial in Uganda. This is my co-chief investigator, Wilberforce Tushamawira, luckily known as Tush. And this is was our project manager from Gates. So again, really very bright orange bananas coming out of this trial. But when we started to do the analysis, we suddenly found that it was even better than we'd hoped. So this is probably our best M9 line. This is the control here. This has got over 40 micrograms per gram dry weight equivalent, so double what we were after. when we went to Nakatembe, we got even better results. So this is our world record at the moment. It has around about 85 micrograms per gram of beta-carotene equivalents. So it's over 4 times the level that we need. And again, we've taken this with no yield drag. So where are we now? So we've just selected the 10 lines that will go into what's known as the regulatory multi-location field trials. And these are going to be planted in Uganda. We're hoping in July, but maybe out to August. We're just waiting on our final approval from the regulator in Uganda. So the planned release now is 2021. We've got to take 3 years in the regulatory field trial and then go through the deregulation process. So what's the outcome? Okay, so our initial target is Africa and Uganda initially. But of course, in those surrounding countries, bananas are an incredibly important part of the diet, including particularly in Western Kenya and Rwanda, Tanzania, Burundi and the Congo. We started in India in 2012, and we've now progressed again using the same concept. We've progressed to field trials in India. A little bit different in India because the field trials are really just a very, very large glass house. So the target population is 125 million people in this region that are dependent on bananas as a major staple food. Fabulously, last year our phase 4 was funded by the Bill and Melinda Gates Foundation again. That will take us through to farmer release in 2021, which is looking like we're absolutely on target for that. Lots and lots of people involved. Obviously QUT, myself and Tush run the program. Now the big group of people is now in Uganda, but that's where most of the activity is. We've been funded by the Gates Foundation, but also the UK Department for International Development. And we're very lucky in 2012, Bill and Melinda came up to North Queensland. They were here on holidays and they came and had a look at the program up there. And I think what was really interesting is, and we spent 3 or 4 hours with them, is at the end of it was one of the scariest questions I'd ever had. So we started in 2005, this was 2012, so we've been going for 7 years. And Bill said to me, "Now James, when will you release the bananas?" I thought, ooh, this is not good. And I said, "We're looking at 2020, Bill." And he said, "Lucky we started when we did." So thank you very much.